All right, so we are finishing up chapter 9. This is part 2. If you forgot to watch part 1, go back and watch part 1. This is plant growth and development. So we are talking about cell division here, not going to go super in depth. You guys have studied this in biology. If you haven't taken biology yet, you will study this. So when you talk about cell division, there's a few processes that are brought up. The most often confused, I would say, are mitosis and meiosis. Mitosis is asexual reproduction in plants. Basically, it can be done with one plant. Also think of it as cloning. We will talk about plant propagation, where you can actually take and cut a piece off of a plant and make more plants. It's kind of cool. You're basically cloning it. So through mitosis, there are steps a cell undertakes to duplicate and divide and complete a set of chromosomes. So if you look at this process, basically, mitosis, you're taking one cell and making two cells that are exactly the same. If you look at meiosis, it's very different, and that is usually what's associated with the whole, sex cells and reproduction, especially when you're talking about people or animals. Plants are very different. Cytokinesis, that follows mitosis and is the division of the actual cell into two new daughter cells. So let's look here. This is meiosis. So in meiosis, when you get through all the phases, you're going to have 4 cells at the end. And it's a nuclear division, but rather than replicate the number of chromosomes, the chromosomes are divided in half. So just like you already know, you get half of your genetics from your mom, half from your dad, and that's how some plants are going to work. And then if you look, fertilization, that's the male gamete that combines with the female gamete to create a genetically unique organism. And that with plants, you're going to be talking about pollination in that realm. So it's a little bit different, and we'll get into that more too. So chromosomes, we're not going to get into this as much. Just know that each plant carries its own genetics, and that it can, pass tributes on. When it talks about those different genes, basically every plant's carrying genetics, and it's going to have a certain DNA structure that's holding information that makes it unique. So each plant, hopefully when you get, like if you're looking at a vegetable and you want a bell pepper plant, and you think that, oh, that plant and that fruit looks good, it looks nice, it's a sweet flavor, and you want a plant just like it, they work hard to make those genetics happen so that when you breed a plant, it's going to show up with those same genetics each time. That's kind of that big takeaway here that I see with this for our purposes. And it's showing you meiosis 1 and meiosis 2. So there's 2 phases to that whole process before it gets to that sex cell that's going to have half of the parent's information. So fertilization, this is kind of the interesting part too. So gymnosperms, those are pollen-bearing male gametophytes. And that, if you look at this, it's showing you in the picture, it's a female cone of a Douglas fir. And those are going to be ovule-bearing scales. So if you see these, some plants are kind of goofy where these will be sealed up. Some are going to pop open and their little seeds will pop out. These are basically the little seeds in here. And the beautiful part is plants are trying to produce more and spread their species. So they're hoping that animals will carry them. The wind will carry them. Some of us have all played with those spinners that are on a box elder tree. They kind of, when you drop the seed, it kind of makes a helicopter spin to the ground. Angiosperm, those are the pollen-bearing anthers. The female has the stigma. So if you are looking at that structure, which we will do something else with that structure so you guys can see it too. But there are, the pollen is going to be grains. Usually they're like a yellowish color, and some plants, especially like Easter lilies, they can stain things, so they actually remove them before you get them sometimes, unless the bud isn't opened. But that pollen has to get on the stigma in order for that plant to be fertilized, and then it's going to produce more of that plant or produce a fruit, depending on what type of angiosperm it is. So the guy that is credited with all this stuff is Gregor Mendel. He was a monk. He did a lot of research with pea plants. That's why it's got the picture of peas on it. He noticed that through working in the gardens that different pea plants had different characteristics. And he started to play around with these plants and figure out that through, pollinating with different pea plants, he was getting different results. So he did some different plant breeding methods, and he's really credited with genetics as we know it today. When you look at this too, they talk about homozygous, heterozygous. This is your old friend, the Punnett square, and if you haven't learned about these, you will. What it's trying to show you is those capital letters on here are going to be the dominant traits. Those lowercase, they have Y's for the letter, which is sometimes hard to see, but if you look here, that lowercase y is the recessive. Usually what happens is a dominant is going to mask the recessive. So in this scenario, it looks like they're comparing color. So if they had one parent, actually both these parents would be called heterozygous. They have both the dominant and recessive trait. The dominant trait is going to mask the recessive trait. So you can see that in all three here. And then this little guy here has the double recessive. So it's homozygous green. It's going to basically anything that is bred, if it has this characteristic here, it's going to breed only green because it only has the recessive trait. This one would breed only that yellow color that it's showing. These two are just like these parents where they're carrying this green trait. And they could have, if you look at this, 25% of the time, it's going to be a green plant. That's kind of your genetics in a nutshell. We're not going to do more with Punnett squares in here. It's to give you exposure to it. So plant breeding, that whole dominant recessive is important to know, especially if I'm going to have these petunias and I want to make sure I get a pink flower each and every time. I want to know if that's a dominant or recessive trait, depending on what I breed it with too. If it's a recessive trait, I'm going to have to make sure to breed it to a recessive plant. There are some instances where it'd be an incomplete dominance, where if I took this pink petunia, bred it with a white petunia, maybe I'm going to get a muted pink color, maybe a super light pink. And then the other goofy part is we don't know if this is a plant that is the dominant trait. We don't know by looking at this. What you're seeing is actually going to be the phenotype. That's the physical characteristics. So, this pepper here, that's red. That's the physical characteristic. The piece we don't see is the genotype. We cannot see if it's a carrier of a different trait that's not visible to the human eye. Plant breeding process, you've a female parent plant. There's different, the hard part that it's not showing you on this slide. You can have cross-pollination. This is kind of what that image is showing you. There's two different flowers bred together. They may not always be the same color like this picture shows. Some plants, there are some different sunflowers even too that have a female plant and then a male plant. And you can actually plant them in the same field and then do some crossbreeding that way. Lots of different situations where that can come up. And plant mutations. Mutations can happen naturally. This one's actually showing a tulip that has a yellow mutation on the side of it. It could be genetic changes that randomly occur. It could be something that if you were to breed this tulip, it could show up in the next generation. It could also be a reaction to chemicals or some sort of radiation that occur. Most mutations are undesirable. Some examples of mutation that is undesirable, it could be lack of chlorophyll. It could be a unique growth habit. Sometimes we encourage these mutations and cause plants to have unique growths. It says chimeras. Those are mutations that allow two genetically distinct tissues to coexist. And then the sports. That's tissue mutations that occur in vegetative cells, for example, a branch or stem that will show specific observable trait differences. It could be variegated leaves or flower color. This variegated leaf is actually, they either call it a snake plant or mother-in-law's tongue. And it can, depending on how it's cut, can sometimes have different color variations, because this is a plant you can actually cut a section off, stick it in soil, and it will grow. It'll start growing roots. Plant physiologists, this is the career connection piece that they have at the end of these. Plant physiologists, they understand plant processes. They examine physical, chemical, and biological functions of plants. Basically, they know and understand how plants work. And honestly, a lot of these say bachelor's degree. It really depends on what you'd like to do. Physiologists do tend to work a lot in lab settings as well. And then a plant breeder, they improve plant characteristics. The traditional processes and biotechnology are used. There are a lot of, like it says, works at university or private companies. This could be in a lab. It could also be in a field. There's a lot of different opportunities where you actually get to do field research and be outside. You don't always have to have a graduate degree. I actually did some work, not as a plant breeder, but I worked for Bayer Crop Science one summer. And, I was an intern. I learned a lot. It was a great experience. I think if I would have continued with them, they would have kept me on as long as I'd gotten my bachelor's, I would say. So lots of opportunities out there, and I hope you enjoyed part 2 of Chapter 9.